ये ऊपर वाला भी जो है ना थोड़ा डिम करना, उनको बोलता होता है इतना।
Hello. Hello, everyone. Good afternoon, and welcome to yet another public lecture, which is hosted by ASET Collegium and Department of Biological Science. I'm sure this topic is very interesting, and we are going to have a very great lecture from a prominent scientist who is here to deliver a public lecture. I request Professor Vidita Vaidya to please come in and introduce the speaker. Thank you.
Thanks, Parag. So this is a joint ASET colloquium along with a public lecture hosted by the Department of Biological Sciences at the Tata Institute of Fundamental Research. So welcome. We often have public lectures, and our public lectures are hosted in this forum. We'd love to see more of the city of Mumbai coming in regularly for these public lectures. I mean, it's nice to see that we're beginning to see the crowd coming in. I know hopefully we will fill those seats. So we'll see in the next half an hour or so. I'm also trying to slow down my introduction so that people have a chance to come in and sit. It's a real treat to have Professor John Cryan here with us today. John is an expert stress neurobiologist, converted over time into being a gut microbiome person as well. John is professor and chair in the Department of Anatomy and Neuroscience at the University College Cork in Cork, Ireland. He did his bachelor's in biochemistry in Galway, and that's where he grew up. And then he did his PhD at Galway as well in pharmacology. Following that, he jumped across the pond to the US, where he did two postdocs, one with Irvin Luckey at University of Pennsylvania, UPenn, and then his second one at Scripps Institute. After a stint at Novartis in Switzerland for a few years, he then joined as a faculty at University College Cork in 2005. He has been at Cork since at UCC, and he's now Vice President for Research and Innovation at University College Cork. He heads all of research at the UCC campus. He's also a principal investigator in the APC microbiome in Ireland, and he has published over 700 peer-reviewed articles, received numerous awards from the European College of Neuropsychopharmacology, from the European Behavioral Pharmacological Society, from the British Association of Pharmacology, from the Physiology Society, American Gastroenterology Association, Neuroscience England, and also FASEP. He was awarded an honorary degree from the University of Antwerp in 2018, and he's been on the highly cited researcher list in 2014, and then from 2017 to the present. I am not going to embarrass John by declaring his H index, but those of you who are curious may want to take a look. It's an unusually high one, and it's reflective of the impact of the work to diverse disciplines, from stress biology to neuroscience to gastroenterology to fundamental understanding of resilience of body and brain in coping with stress. He's an elected member of the Royal Irish Academy in 2017, and he co-wrote a book with Scott Anderson and Ted Dinnan. I think it was written in 2017, I think, if I'm not mistaken. It's called Psychobiotic Revolution. It's written for the layman. It's a remarkably interesting book because it talks about what we perhaps didn't fully appreciate, which is that most of our body is not really our cells, but the bacteria that coexist with us and how they shape our body, brain, and behavior. It's my absolute pleasure to introduce Professor John Cryan and to have him come up here and give the public lecture. I'd also like to invite our Dean, Shankar Ghosh, to say a few words before that, and then to welcome John and hand over to John as well. Shankar, you want to come up?
Thank you, Vidita, and thank you, John, for visiting us. And it's great to see people across the city to come and visit us here. And we hope that this exercise will continue and we'll have more of the Mumbaikas visiting TIFR, listening to what we do, and participate in the excitement that we all share amongst ourselves. I was just talking to John now, and John is from Ireland, and India and Ireland goes a long way about. We share a close history during our colonial eras, and John promises that he is going to come next time to TIFR on a proper visa and is going to spend some time here with us, not a short one, like a half-a-day visit, but going to be here for some longer period of time. Now I see that people have almost come in. So yes, Sean, I invite you to come up to the stage.
Okay. Thank you so much to the Dean and for everyone for their welcome here, with a special thanks to my friend Vidita for inviting me here. We know each other for a long time, and it's my first trip to India, so it's a real pleasure to be able to come to TIFR and come to Mumbai and to share with you the journey that we've been on in Cork, in the very south of Ireland, over the last almost 2 decades. This year will mark my 20th year returning home to Ireland, and I never worked on the microbiome until I moved back to Ireland, where I started collaborating with some cool people who started asking, well, what if? And then we allowed the data to take us to this place. And it's been really remarkable for me to watch the field grow, to watch people start to understand that basic science can lead to translational science, can lead to actual changing practices for every day for people in society. And I understand that we have a mixed audience of people who are just interested in the whole concept of gut feeling. So I'm going to take you on a kind of a whirlwind tour, and I'm happy to take questions thereafter. Okay, some geography. I always like to start with geography. Most people in this audience will be able to figure out where Ireland is overall. The most important thing to know is that we're not part of the United Kingdom. We're not united or a kingdom. So this audience will appreciate that. As we just heard, we do share many links with India over our pre and post colonial lives overall. But what people mightn't know is the map of Ireland. And this is really all you need to know about the map of Ireland. Here is Cork. It's our biggest county, but our second city. Here it is the place where the Titanic stopped for its last trip and where the Lusitania got bombed off it. So, and for many people in India, it's also known for where, unfortunately, the Air India accident happened in 1985 as well. And there are strong links between the communities in Cork and the families of the people in that tragic accident overall. So I work at University College Cork, which is 180 years this year. We, like your Mumbai, love our neo-Gothic architecture. But also, I just wanted to highlight that University College Cork is home of many amazing Indian researchers, scientists, students who come from all parts of India and have a very vibrant society on campus in Cork as well. And we like to keep that alumni going. All right, so on to my story. This is a painting by Matisse, and I like to tell the story because it's a painting that hung in the Museum of Modern Art in New York, just like this, as part of a retrospective exhibition in 1961. And everyone came to see it at that time, including Matisse's son. But they didn't notice something. And it wasn't until a very eagle-eyed stockbroker called Genevieve Habert came by, and she noticed that something was wrong. And what was wrong was that the painting was hung upside down, and people were looking at it the complete wrong way around for all this time. So, if you take nothing away from my talk today, except to remind you that sometimes we have to turn things upside down and look at them in a different way to really understand how they are, then we can really appreciate how the brain is. And I am a neuroscientist, so I have the primacy of the brain in everything that I think about overall in all of my training, but I want us to think a little bit different. And the concept and I work in a medical school, like many people here, I assume, or have been trained in medical schools. And in medicine, everything goes back to Hippocrates, who's thought to have said, now it could be fake news, but he's thought to have said that all diseases begin in the gut. Now, when I talk to my neurology or my psychiatry friends, they would challenge this very much in terms of how we are. And my neuroscience colleagues, who are by far the most skeptical of all, often challenge me and say, John, why, if you're interested in the neurobiology of emotions, which I am, why are you so fixated on the gut? But all we have to do is look to our language. We use phrases like gut feelings and gut instincts. We're gutted when we're disappointed. We make gutsy moves when we're brave. We have butterflies in our tummy when we're nervous. So we do personify our emotions deep in our gut. And what we've been trying to do is try and understand what this might mean from a biological perspective. Now, over the last probably 2 decades or so, there's been huge advances in technology that have allowed us to start to really understand the microorganisms that we have in and on our bodies. And what that has allowed us to really to grapple with is that we are living in a microbial world. Microbes were here long before humans. And it is really, really humbling to think about it. And when we talk about microbes and the microbiome, these are the trillions of bacteria, viruses, fungi that are living in and on our bodies. And I'll share with you some of the numbers because they are quite humbling. If you looked at the genes that we have in and on our bodies, we would be more than 99% microbial. All of the money we spent on the Human Genome Project, and it's for less than 1% of our genes. The weight of our gut microbes is roughly that of the brain, which is very humbling as a neuroscientist. We have roughly the same amount of microbial cells as we do human cells. And so as I like to remind people, just think the next time you go to the bathroom and you get rid of some of your microbes, that you're becoming more human and what that might mean overall to what you're doing. And grappling with how these microbes are playing a role in our overarching health and how they may play a role in disease has been a big area. But really what I wanted to reinforce also is that it's so important that we put this within an evolutionary context. Yes, the microbes were there first, and they have co-evolved. We have co-evolved as humans with them. So this is a concept known as the hologenome concept or holobiont concept. And so together, these microbial and human genes have to navigate an environmental metagenome. And once we start thinking about this from an evolutionary perspective, then we start to really have a different perspective on the role that these microbes might play. And what we've been interested in my lab is understanding the relationship between the microbes and the brain at key windows across the lifespan, in early life, in adolescence, and all the way into old age. And I'll briefly talk about all three time periods today, but I won't have time to go into any great detail. Now, where does it begin? Well, for the most part, we're thought to be sterile in utero, but we get our microbes as a kind of a birthday present from our moms on the day we're born. And these microbes, these frontier bacteria, they start to inform the developing immune system. They start to help program other systems in the body overall. And so that we're beginning to realize that within that first 2 years of life, factors that will change the composition of these microbes can have potential effects, negative effects on overall health. And these factors vary from mode of nutritional provision, so whether an infant is bottle-fed or breastfed. Host genetics plays a key role. The exposome, the environment we live in, exposure to antibiotics or other medications, and other lifestyle factors will influence the microbes. One of the other factors is mode of delivery at birth. Because what we see is that in infants born, and this goes back to classic work from Gloria Domingos-Bello's group, where she looked at a cohort of infants who were born by cesarean section and compared them to those born vaginally in relation to their microbiome composition, and she found that they were quite different. And it's like if you compare this transmission from mother to infant as a kind of a evolutionary relay race in life. If you start to see that, then what happens in C-section, the infants start picking up the bacteria from the hospital environment or the skin. Therefore, the baton in that relay race gets dropped. And the microbes are no longer the security blanket. One of the largest studies looking at this is the Teddy study in Nature some years ago, which again reinforced that C-section is one of the key factors in early life to drive these changes in the microbiome. Now, epidemiology studies tells us that infants born by C-section are at an elevated risk of allergy, asthma, and type 1 diabetes. It doesn't tell us much yet about it in the context of brain health, although it's something that we have been collaborating with our epidemiology colleagues with over the years. As an experimentalist, I've been working on animal models to look at this, and we find in mice that are born by C-section that they have enduring effects on brain and behavior. And the good news is that we could actually reverse some of these by reversing the changes in the microbiome in early life. But in relation to, we wanted to test whether we could translate any of this to the human side of things. And to do that, then we decided to take, and I was going to say normal, but Irish medical students, and stratify them based on their mode of delivery at birth, because most of them know whether they were born by C-section or not. And then we bring them into the lab, and we have very good and ethical ways of stressing people in the lab. One of them is they come in and they have to pitch their dream job in front of these very grumpy looking people in white coats. And then they have to make a pitch. After they made that pitch, they have to do a mental arithmetic test they don't know anything about. So count back from 2017 in 29s, or something like that. And they'll make mistakes and then they have to start again. And we're measuring everything along that line. And what we find is that individuals born by C-section have a increased immune and psychological response to this acute stressor. Now, acute stress is interesting and overall, but I'm interested in disorders that are more associated with chronic stress, anxiety and depression and et cetera. So is there a way then that we can start to look at this in an ethical way in the lab? And that's what's really good about being an educator. We do it all the time by subjecting our students to examination stress. And so we can take advantage of this and look at how students are responding across the exam period in both semesters. And we found again that individuals born by C-section have a modest but significant immune and psychological, as measured by the Cohen's perceived stress scale, response to stress. Now this was interesting because it built on our animal work and built on some early epidemiology work and does tell us that the microbiome might be involved, but it would be in no way convince my very skeptical neuroscience colleagues. So we have to come up with more evidence that the microbiome is relevant for brain and behavior. Because this is, if I took you back 15 years ago, this would be seen as something completely out there, something completely that this crazy guy in Ireland is talking about, but no one else was really believing at that time. So I needed to get more evidence. So how do you get more evidence that something is important in a process? Well, all you have to do is then look to engineering or other aspects of biology. And what we do is we take it out and see what happens. And so the concept of growing up without microbes, growing up what we call germ-free, isn't new at all. It goes back to Louis Pasteur's time. Here's a germ-free man from a sci-fi magazine from 100 years ago. He doesn't look that great overall. We work with germ-free mice. Now, while they have their own problems, they do look a lot better than he does. And these animals have allowed us to answer the question, is the microbiome involved, yes or no? And are a starting point for our programs of research overall. There's no real human translation, with the exception potentially of the boy in the bubble that Paul Simon sang about in the 1980s. But these mice in the bubble have taken my lab to all kinds of weird and wonderful places that I would never have predicted that we would end up going. Places like showing clear changes in amygdala development. The amygdala is the brain region very much involved in social behavior and fear learning. And here you can see this is a brain cell, a neuron from the amygdala, and you can see that it's in this what we call arborized, tree-like state already in germ-free animals overall. Myelin is a very important part of our machinery in neuroscience for appropriate conductance. Myelin is basically the insulation on brain cells that allows them to conduct appropriately. And we mainly think of myelination in the context of demyelinating disorders like multiple sclerosis, where the myelin gets attacked by our immune system. Now, we looked down the microscope. We first showed that there were genes involved in myelination were upregulated in a region of the brain involved in higher cognition, the prefrontal cortex, in these germ-free animals. And then when we looked down the microscope, we found that there was increased myelination, hypermyelination in germ-free animals. Now, hypermyelination, increased myelination, doesn't occur too often in nature, with the exception of some rare autisms. So this was kind of surprising to us. But what was most remarkable for me was that we were able now to see that by targeting the gut microbes, we could affect a fundamental process in our brain that's so important for brain activity and conductance, and really opened up a whole new world. We've now reproduced this more recently in zebrafish, and to show that there's evolutionary context to this, and we're looking at the mechanisms at play. Other processes that we've shown in germ-free animals, we've shown that adult hippocampal neurogenesis, the birth of new neurons, that this is out of kilter in germ-free animals, and changes in plasticity-related proteins has emerged. And what does that translate to in terms of behavior? That's always what I'm always thinking about as a readout. And mice, like humans, are quite social. So if you give them the chance to spend time with another mouse or an empty chamber, they'll gravitate towards the mouse. but not if they're germ-free. And mice, like maybe some humans, are a bit fickle. So if you give them the chance to have a new playmate versus their everyday playmate, they'll gravitate towards a new playmate. But again, not if they're germ-free. So fundamental aspects of sociability and social cognition are gone awry when we don't have appropriate microbes in our gut. And that for me was a real eureka moment, because it tells us that there's something about the social brain in particular that makes it sensitive to these signals. So then I collaborated with a zoology colleague and an evolutionary biologist, and we started surveying across the animal kingdom where is the evidence that when you disturb the microbiome, what is the type of behavior that is most often reported? And that is changes in social behavior. And vice versa, that if you start to change social hierarchies or social dynamics, whether it's a baboon in the wild in Africa or a bumblebee in a hive, you start to really change the microbiome. And so we're beginning to see some sort of clarity on that. In relation to disorders, a lot of the focus in the field has been on in the context of complex aspects of sociability related disorders, particularly related to autism. We've been more recently more interested in another disorder called social anxiety disorder, very common but not very appreciated disorder. It has a lifetime prevalence of 12%. It's probably the only disorder that got better during the pandemic. And it's a disorder characterized by that crippling feeling of being judged, and where people are just not able to in public situations. And it has a very distinct biology, but it's not very well understood. And so we were interested to see, because of the role of the microbiome in shaping the social brain across the animal kingdom, could it have a role in social anxiety disorder? And my clinical colleagues, we investigated this, and we did find that we could correlate the changes in the microbiome with disease severity in individuals with social anxiety disorder. But that was correlation, so we needed to go one step further, and therefore Nate Ritz in the lab at the time, a PhD student who's now back in the US in Seattle, decided, well, what if we took the microbes from the individual who has social anxiety disorder, compared it with the microbes of those who don't, and gave that to an animal model, a mouse model. Could we induce social anxiety disorder in a mouse? And this was really one of these kind of crazy ideas that panned out. And we were able to, in collaboration with David Slattery, a former postdoc, we're able to show that normal, healthy mice that get the microbiome from patients with social anxiety disorder, that they have a deficit in what we call social fear extinction. So they're not able to learn to have appropriate social interactions with another mouse, compared to mice that get the microbiome from healthy controls. So again, reinforcing that there is a clear link between the social brain. And so when all of this data started coming together, I started having these conversations with the postdoc in the lab at the time, Roman Stilling, and we started saying, well, now we can start piecing this triangle together. The microbiome is important for brain development. Microbiome is important for social behavior. We know from Robin Dunbar's theories that social behavior is really important for cortical development and for elaboration of the social brain. So we started to think about this. And one of the things that we began to see is that, and ask the question, what is in it for the microbes? Because here in this very crowded auditorium today, all of you have an aura of microbes around you that you are now nicely passing on to the person beside you. And so being in a social environment allows for perhaps microbial fitness. Whereas those of you on YouTube or listening on YouTube or on Zoom or in your own little private your home, and you're keeping your own microbes to yourselves. Thank you very much. And so we started coming up with some of these ideas about what's in it for the microbes. And then I put these to Seth Bordenstein, an evolutionary microbiologist, and he told me that I was completely wrong. And every now and then, it's really good to be told that you're completely wrong. And why am I wrong? He told me I was wrong to even be thinking like that or asking that question. And the reason is that because the microbes were there first. And there never has been a time where our brain has existed without these microbial signals. And we have co-evolved with these friends, and these are friends with benefits, and these are social benefits. And so that changes where we are overall. And we began to start to understand this very, very much in relation to all of these factors that I mentioned earlier, how they can impinge on this. And maybe, and this is provocative, maybe we can, by tinkering with the microbiome, through these various mechanisms, that we can start to influence the social brain. We can start to change empathy responses. We can make people work better in teams. We can make, the Irish cricket team win again. We can start to develop trust in each other, and we can cooperate in a different way. If that was true, and we would all be sending big boxes of such treatments to the White House, but if it was true, it opens up a whole new provocative idea for normal social behavior that we would be able to really target it through what's going on in the gut, and that really the main social network isn't Facebook or Meta, but is your microbiome overall. We still have some way to go to get to that, but it is really important. And one of the key things that we're seeing time and time again, and Minal Jagger, who came from Vidita's lab to us some years ago, and I'm delighted to see he's here in the audience, wrote this really great review to really highlight one of the other facets of this story that I'm going to put my hands up and say, I don't know why, but it's just the case, is that there's something about the male brain that makes it more sensitive to these effects, or something about the female brain that makes it more protective overall. But we see this time and time again in our animal studies. And really understanding this is going to be an important area for the future, looking at sex differences because of the sexual dimorphic aspects that mature in relation to disorders of the social brain, whether it is autism, adolescent onset schizophrenia, or social anxiety disorder overall. But what we're really now beginning to see is the concept of critical windows, which is an important concept in neuroscience in general, where we know that there are critical windows early in life that are important for socio-cognitive development, that there is also similar critical windows in our microbiome. And what we're trying to do is now juxtapose these together to try and really piece to understand how we'll move forward in understanding this. And there's a lot of really exciting work in early life trying to understand that overall. I'm going to move quickly to adolescence. This is really an important area because the adolescent brain is still very much a brain under construction. It is the last stage where neurons are basically being pruned, and we have basically interactions between immune cells in the brain and the neurons to prune off the cells that we no longer will need into adulthood and overall. And we know that factors ranging from alcohol, stress, poor nutrition, inadequate sleep, and drugs of abuse can all affect that adolescent brain quite remarkably. But we know less about what they can do to the adolescent gut and in relation to it. We also now appreciate that adolescence doesn't stop at 18 or 19. It goes on into early 20s. To college age overall in relation to the dynamics that's happening in the brain. And so we were interested in this and have been overall, and we've shown that in animal studies that if you wipe out the microbiome during this critical period, it has enduring effects well into adulthood overall. And it also allows us to intervene with positive diets during this period as well. But then one study I thought I would share with you today is a study that Karina Cariba did in our lab. She was a Spanish a psychologist who came to Ireland to study binge drinking. I'll just leave that there. That might sound like a good idea until you start needing controls in your study. And that was a problem with this study as well as COVID. But one of the things that we found, and really, again, reinforcing our overall thematic today, is that there was microbiome signatures of binge drinking and craving that would allow us to be an early biomarker of future relapse, etc. We were looking at relationships also between higher cognitive readouts, especially social cognition. And again, the social brain came up in our studies overall. And so we're beginning to see a very close relationship at this critical time window between alcohol consumption, rewarding pathways in the brain, as well as social brain, and how the microbiome is driving these. And this is something that has received some attention in the press overall, and may offer some potential for helping people who struggle with binge drinking, etc. And again, we're seeing this more and more, the gut-brain signaling. There's papers just out on GLP-1, for example, on alcohol abuse in the last few weeks as well. So we're beginning to see a more holistic view on this overall. Okay, now I'm going to switch to the other extreme of life. Here is one of the heroes of the story. Does anyone know who Eli Metchnikoff is? Up hands if you do. And if you do, then I will know how many immunologists are in the audience. So Metchnikoff he was a Russian Jew who grew up and was educated in actually what's now Ukraine, in Kharkiv. And he did most of his seminal work at the Pasteur Institute in Paris. And he discovered the process of phagocytosis, which is basically how immune cells gobble up foreign bodies. And he was a polymath. He was interested in so many different things. But when he turned 50, he started getting worried about getting old, and really started writing about, and he was full of crazy ideas about everything, overall. But then he got fixated on aging, and what could the secrets of healthy aging be? And he started looking around Europe to see, where do people age healthier and better than other places? Because the center of Paris mightn't be the best for your age overall. And what he put it down to was places like rural Bulgaria. And he wrote about it in this book called The Prolongation of Life. And people in villages in rural Bulgaria, he sought after, were drinking a lot of fermented beverages containing lactic acid bacteria. And so he put that as one of the key factors for healthy aging. So he's not only the father of immunology, he's a father of geriatric science in a way of doing natural history studies, and the father of probiotics. So if you go to Korea, you can get a yogurt drink with his picture on, and it must have that Nobel sign of approval and must be good for you overall. But basically, because he had so many crazy ideas, all of this was forgotten about for at least another 70 or 80 years. But now, over the last 2 decades, people have begun to realize the clear relationships between these lactobacillus and various bacteria in the gut and healthy aging. A study published over a decade ago now from my colleagues in Cork showed a clear correlation between age-related cognitive or age-related frailty and the composition of the microbiome. And diversity is really important in all aspects of life. And what they showed there was that diversity was really important in your microbiome. And the individuals who aged healthier had a more diverse microbiome, and they put it down to what they ate, which was having a more diverse diet. So that was really what the take-home message of that paper was, and of the field moving forward. But it has clear public health ramifications, because in Ireland, in our nursing homes, we tend to give our individuals who stay there a lot of beige and bland food, and that's not what's good for their microbiome, and that's not what's good probably for their overall health. As an experimentalist, we started revisiting Metchnikoff's ideas in our mice, and we showed that aged mice have changes also in their microbiome, which will correlate with changes in behavior and inflammation. But this is a correlation. Starting point, but a correlation. How do we move forward? And so one of the ways to move forward was to say, well, what if we gave a diet that was good for your microbiome? We call it a prebiotic diet. And so we decided then what if we took older animals and gave them a diet that was good for their microbiome and see did it slow down any of the effects of aging? And for this, I decided to focus on middle age. For obvious reasons, it's a time window of interest to me. And so we could see already in middle-aged mice that by 11 months, that there is already the portents of cognitive decline in their brains occurring. There's increased infiltration of inflammatory monocytes and increased microglia activation. These are immune cells. So already what we call neuroinflammation is starting to occur in these animals. And what Marcus Boehm, the postdoc in the lab, did was feed the animals a diet rich in inulin. Now, inulin is found in many vegetables. Among them are leeks and chicory and artichokes, and I'm sure many in Indian cuisine too. And in the animals that received the inulin, we were able to have a diminution of the effects of middle age already. So this is telling us that feeding our microbes was really good for slowing down the effects of middle age. But diet, and also the other part of this story then is that it tells us and reminds us that if you're thinking of having a midlife crisis, maybe you should forget about the motorbike and turn to the leeks instead. And that might be a better scenario. And these are my clinical colleague, Ted Dinan, and my close colleague, Harriet Shellikins, who worked on this project overall. But this diet can affect the brain directly. It doesn't have to go through the microbiome. So we couldn't really say that this was definitely a microbiome-mediated effect. So then I convinced Marcus to say, I said, Marcus, if we believe that the secrets to healthy brain aging lies in the microbiome, then we should be able to take microbes from young animals and give them to older animals and reverse some of the effects of aging. And by the way, this isn't going to work. That's what I told him. I said, it's not going to work. And but he took on the challenge, and indeed that's what we did. We took the microbes from 8-week-old animals and gave them to 22, 23-month-old mice. These are geriatric mice, and remarkably, we were able to show that we could reverse the effect of aging on the microbiome, on peripheral immunity, on brain immunity. When we looked at the chemical composition of the hippocampus, the region of the brain involved in learning and memory, it was completely rejuvenated overall, and changes in behavior related to cognition and anxiety. And this for me was very exciting. And it has been reproduced now by 3 other groups since, which is really encouraging overall. Now, the take-home message here isn't that you should all run out and start stealing your young kids' or grandkids' poop. That's not what we're trying to advocate, but it gives us a proof of concept that we need to mind our microbes to be able to look after our minds as we age overall. And what we're trying to do now in our lab is trying to figure out how on earth does any of this happen? How on earth? So, in Ireland, being very proudly Irish, we don't have too many Nobel Prizes in medicine and physiology like Metchnikoff, but we have many in literature. And one of them is Yeats, who was probably known most in India for bringing some of the work of Tagore to Europe and Western world. But Yeats is a poet who won the Nobel Prize just over 100 years ago, and he was fascinated by trying to disentangle complexity in things into individual components. Whether it's a chestnut tree, whether it's the leaf, the blossom, or the bowl, or how can we understand what the dancer from the dance? And so that, to me, is really what we're trying to do in my lab right now, is trying to understand this complexity of human homeostasis in a new way. And I have a really wonderful Indian originally postdoc, but now on faculty in pharmacology in Cork, Jatin Nagpal, who together with we were able to sneak this Yeats quote into this review from Neuron to really highlight some of this complexity overall. And we're just scratching the surface right now. We have a long way to go in trying to understand that. But we have to invest in this. This is too important. This is brain-body science telling us about how we can be healthy in a normal society, and we have to be able to dissect out this so that we can know when and what to intervene with in a science-informed way, and that will give the public the answers. So badly need to be able to inform them about their own health overall. And that's the journey we're on. So we have some inklings about how it could work. One of them is through this vagus nerve. Vagus comes from the Latin for wandering. It's words like vagabond and vagrant come from the same origin. And we showed now in a classic paper in 2011 that if you cut the vagus nerve, all of the effects of a specific bacteria on brain and behavior were gone. And so this reminds us that what happens in Vegas doesn't just stay in Vegas, but will actually affect our emotions in certain ways. And at least helps us know that there is one pathway that this is involved in. What we're trying to do now is what happens when these signals reach the brainstem in the brain? How do they impinge on circuits that are really important for complex cognitive and emotional behavior? And that's ongoing work at a very fundamental neuroscience perspective overall, and it's quite exciting. Also, I like to remind people that the microbiome is like a factory. It's our body's factory producing all kinds of weird and wonderful chemicals that our bodies wouldn't be able to produce otherwise. Among these chemicals, for example, are what we call short-chain fatty acids, really important for giving energy to the cells that line our gut, but also the cells that line our blood-brain barrier, also play a role in signaling directly to the brain. And factories depend on many things, but two of the things that factories really depend on are the raw materials and the quality of the workers. So here, the workers are the bacteria themselves, but the raw materials come from the diet. And that's really important. So we need fiber in our diet to be broken down by these workers to produce these short-chain fatty acids. And they're really important. And so once we start to understand that, we start to appreciate how these chemicals are building up in our gut all the time and how they're getting into our bloodstream and signaling to us overall. Maria Aborto came to my lab as a postdoc, and Maria is fascinated by two things in life, at least. One of which is barriers in general, and the other is the Lord of the Rings. And so in this review here, where we have the Gates of Dorian on the cover of Nature Reviews, she highlights, the different barriers that are really important along this microbiome gut-brain axis. So we have a blood-brain barrier, which is really important. And if this gets leaky, it starts letting things in that shouldn't get into the brain. We also have a blood-cerebral-spinal fluid barrier that's really important for maintaining brain homeostasis and vascularization. And at the gut, we have a gut barrier. Now, people often talk about a leaky gut. Now, our guts aren't really leaky. It's really not a great term to use, but the integrity of these barriers can get compromised and allow things to get, especially bacteria, into places they shouldn't get to and cause damage. And so that's another area of research we're really interested in. How does that play a role in how microbes signal to the brain? You mentioned, Vidita mentioned the book we wrote called The Psychobiotic Revolution, which comes from this term psychobiotic, which is a term we came up with in Cork, Ted Dinan and myself, means targeting the microbiome for mental health benefits. There are many ways, many of which we've already talked about already, specific strains of bacteria, combinations of bacteria with prebiotics, these dietary substances like inulin, fermented foods, etc. But revolution is a very strong word. We're probably at the beginning of some sort of revolution. But one thing I can say is that as someone who works in a medical school is that we need to also start at education. And our doctors do not get enough nutrition advice in their training. Our dentists get more. And so we have to really try and integrate nutrition and clinical nutrition, science-based, into understanding how we may help and manage certain diseases and prevent others from happening overall. I've been excited in to see some of the strains of bacteria that we've shown in animals to have positive effects go into humans. This is a psychologist, Andrew Allen, in our team, and we're able to show that in healthy volunteers, we're able to show that they can modulate brain activity. Work from Germany, from Paul Eng's group, showed in a brain scanner, you put a person into the brain scanner and they're playing a game in virtual reality with two other people, and they're passing a ball from one to the other. It's a very nice game. And all of a sudden, the person in the scanner gets excluded. Your brain doesn't like to be excluded. It takes it back to all of them playground overall, and it mounts a stress response. But if they'd taken this bifidobacteria, then it didn't do that. And that's quite kind of cool. But not everything comes through. This is a study where we looked at a strain that we showed was vagal dependent, and it didn't do anything in our human studies. And so this is always the challenge when we're moving from basic science up to humans, that there are hurdles for us to go, and it's not specific to the probiotic field overall. But time and time again now, we're beginning to see, when we put all these studies together in various systematic reviews and meta-analysis, we're beginning to see that there's a clear relationship between food and mood overall. We're also beginning to see this in other areas. The entire sleep field is beginning to wake up to the fact that there's a microbiome. And we showed, again in students going through exam stress, that if they'd taken a bifidobacteria longum, that their sleep quality improved overall. The mechanisms of how all of this is happening is a really exciting area right now. We've been interested in circadian rhythms because in mental health disorders, they're often associated with sleep disturbances and very much associated with disturbances in sleep, in circadian rhythms. And talking to my clinical colleagues, if we could, do something with the sleep of patients in the first instance, it could be the first inroad to getting them towards being well again in certain areas. And so recent work in our lab that we're very proud of, that just came out in the last few months, led by a very talented Brazilian PhD student, Gabriel Tufani, we've shown for the first time that there is a intricate relationship between how our brain and our body integrate stress responses with circadian signals in the brain, and that all of this is driven by what signals from the microbiome. It allows us to think that targeting the microbiome in the future may help our circadian rhythms realign. It may help me, when I'm dealing with my jet lag in the future, be able to take something that it will affect my microbiome and help me synchronize to Mumbai time away from European time overall. But this is something we're very excited about, and stay tuned for a lot more to come on that overall. But it is a new concept, how the stress system puts circadian rhythms out of kilter, and maybe the microbiome could help bring that back, and is playing that yin-yang on that. And that's something that we think would be of good translational evidence. I want to mention viruses. Over the pandemic, they've got a very bad rap. Really bad. But did you know that within your gut, you have trillions of viruses that are helping to keep you alive? And these are the bacteriophages that are basically keeping the bad bacteria at bay. They are our defense system overall. And they have exquisite specificity for certain bacteria. And so Nate, when he was doing his PhD, together with Colin Hill's group, a bacteriophage group in Cork, we were really excited to see, well, how does this link in with stress? And we showed first that stress is changing the composition of these viruses. And then Nate decided to take the healthy viruses from mice before they underwent stress and give it back to them while they're undergoing stress. And we could attenuate all of the immune and behavioral effects of stress. And this was remarkable. And it opens up an opportunity for very specific treatments into the futures, that we may be actually developing viral-based strategies that might actually alleviate the effects of stress overall. And the last thing I want to mention today is really to reinforce where we are in the context of diet. People have gone out and looked at diets cross-culturally, and you can go to parts of Tanzania, for example, and look at hunter-gatherers, and you see they have a very diverse microbiome, and that might be more to do with the gathering than the hunting, but people forget that part of it overall. But that gives you a kind of a baseline of what our microbiome would be like overall. Then with the introduction of agricultural practices, we can see that there's a diminution in the composition of microbiome diversity. And then with the introduction of processed foods, the stressful lives we lead, antibiotics in the food chain, etc. And this is not just the US, this is also Ireland and India in many cases, that there has been an extinction of microbes that our ancestors had. And the question is, can we ever get them back? What we know is that there's very little inflammatory disease, multiple sclerosis or IBD in these populations. We know less about mental health. but it is something that we really need to look at. And in psychiatry in general now, there's been a push towards trying to understand how nutrition could be used as a basis to improve mental health. And there's more data coming out on every week to support that nutritional interventions can support either medication or psychotherapy to enhance outcomes in patients with mental health disorders. And together with Elizabeth Schneider, a psychologist in my team, we put together this concept that we not only have a gut-brain axis or a microbiome gut-brain axis, we really have a diet microbiome gut-brain axis, and understanding how different diets impinge on the microbes to affect how they signal to the brain is going to be a really important area for us moving forward. And among them, one of the areas going to be really keen is of course, fermented foods. And Ramya Balasubramanian is an Indian PhD student with me in the team who is so passionate about fermented foods and what they can do overall, because it offers a really good and inexpensive way of democratizing potential opportunities to support brain health into the future overall. Kirsten Birding was a nutritionist in our team, and she said she wanted the challenge of, if we believe all of this stuff we get from animals, if we believe the microbiome is important for stress, we should be able to feed our microbes, basically enrich it with fibers. So we really ramped up fibers in a bunch of Irish students who have poor diets and are stress sensitive. And we also increase fermented foods, which are almost non-existent on a good day. So, and what we were able to show that there was a reduction in perceived stress in these individuals. And this is my clinical colleague who helped a lot on this study as well. And we're really beginning to see that fermented food and fiber could be a way forward. We're looking at this from a biological sense right now. We're extending this out for 8 weeks. We're looking at whether it's the fermented food, whether it's the fiber, because there's a growing literature supporting that they have different effects on our immune system and different effects on our overall biology and different effects on our microbiome. But if we put them together, maybe that's where the secret of beneficial effects on brain health will be overall. And the whole talk, I've been focused on my passions, which are mainly around mental illness and psychiatry. But it would be wrong of me to neglect that the microbiome gut-brain axis is also very relevant to neurological disorders. There is such an exciting momentum now in the Parkinson's field, for example, where we now recognize a body first as well as a brain first aspect of the etiology of Parkinson's disease. In Alzheimer's disease as well, a lot of really exciting work going on. Autism is receiving a lot of attention, MS, epilepsy, stroke, etc. So hopefully, as we get more data, we can come up with better strategies to move things forward. But the big issue for me and for the field is, how do we get a causality? How can we prove that the microbiome is involved? We can do lots of correlation studies. Here's one I was involved in where we can correlate the microbiome composition and cognitive performance in young, healthy adults. Really nice correlation, but we have to move beyond that, because you can correlate almost anything. So here you can correlate the divorce rate in the state of Maine in the US with the per capita consumption of margarine. Okay? I'm Irish. We like butter. So if your partner came home with margarine all the time instead of butter, it could lead to arguments, which could eventually lead to divorce. And these are the stories we tell ourselves in the fields. They're just stories because we try and create these narratives. And so in moving forward in microbiome research, we really need to understand what a normal microbiome is. We need to understand the effects of various lifestyle factors. We need to have large-scale translational longitudinal studies with better controls. We need to understand directionality, and we need to understand gene-environment interactions across various cultures. We need to understand that most microbiome research today is done in white Western populations, and there's a whole neglected part of cross-culturally in what we're doing, and we have to fill in these gaps overall. But the good news is that, unlike your genome, which all you really can do is blame your parents and your grandparents about, your microbiome is potentially modifiable. And that gives individuals agency over their own potential health and well-being in a way that we never thought of before. So there are some things that I just want to reinforce is that it's very difficult to know what a normal microbiome is. We are seeing a lot of excitement going on in citizen science projects and longitudinal measuring. There's challenges with this. We don't know what the sample size should be. We don't know what the bioinformatics resources need means people from different disciplines. Mathematicians, computer scientists, biologists, microbiologists, neuroscientists, all these psychiatrists, neurologists, all have to get into a room together. And that is the room where things will happen. And it's an exciting room, but it's not always easy to do. We will lead to personalized diets and interventions and understanding this. We need to understand more how the microbes signal to the brain. There's lots of innovation. There's smart pills are being developed. So they will be able to tell people right to your watch what's going on in your gut, what's going on in your microbes. Do they need more dosa today, or what do they need overall in terms of that? And that's where we're getting to in the future. There's a project out of Stanford where they're going to have a smart toilet. It's going to go down very well in Japan, where they're going to be able to have online measurement of the microbiome overall. I do mention disciplinary barriers because we compartmentalize the body in Western medicine all the time, and gastroenterologists and psychiatrists may not be the ones talking to each other all the time, never mind with microbiologists and others. But we need to kind of think more integratively, more forward. And just here's some aspects of what people can do and take home to themselves from my talk today about how you can have a better psychobiotic life. And it's really about increasing certain things in your diet, if you can. So fibers, vegetables, prebiotics, omega-3 fatty acids. The color, it's wonderful coming to India and seeing the color, but in the foods in particular. But the color in foods, a lot of it is driven by polyphenols, which are really good for your microbes overall. They're present in, I often say, dark chocolate and red wine, but equally in grape juice and onions. So overall, various yogurts and fermented milks and fermented foods are really good, as we alluded to. And then you can start decreasing intake of ultra-processed foods that we know are really bad for the microbiome. Emulsifiers, sweeteners really do it. Aerobic exercise is very good for your microbiome. You can try and avoid stress. Sleep disturbances, jet lag are not good. We know that. Trying to get a public health message about avoiding C-section where possible, but acknowledging that C-section is a life-saving intervention, and it's about supporting that and supporting breastfeeding and minimizing antibiotic usage. And then the one that I regrettably told my family was that having a pet, especially having a dog, is good for your microbiome. So if you take nothing else home, you might take that home from you. So I'd like to thank my amazing team in Cork, who come from all over the world to play and really challenge the system overall. You will notice that there are many from India here, as well as two newbies who just joined from India in the last couple of months, and we have had such a strong and wonderful interaction with people from India over time. I'm going to leave you again with Hippocrates, who's also thought to have said, and again it could be fake news, that let food be thy medicine, and I'll change that to let food for your microbes be medicine for your mind. And then just remember that every now and then, just every now and then, it's good to turn things upside down and look at them in a different way. Thank you very much.
Thank you very much for a wonderful talk. Yeah.
Thanks, John. That was great food for thought, truly. It was too tempting to make that comment. But we have time to take some questions, and there are volunteers. So if people put their hand up, then we can actually, there's one here, and then we will.
I believe the elite athletes have their microbiomes for sale. And is that something that can be, is that something that is, that people should think about to reset their microbiome?
To buy the microbiome from elite athletes. I haven't heard that, but my colleagues in Cork have been studying the elite athletes a lot, and it is quite a different microbiome, and they've been looking at it for also in relation to diet, because elite athletes' diet is quite different. So it's trying to dissect the relationship between the high protein intake and various other things that elite athletes have versus what would be important for their performance. There are some studies in marathon runners where they've taken the microbes from the marathon runners while they're doing like at the end of the marathon, just about, and they found that there was a change in lactate producing bacteria, and then transplanted that into animals and got them to improve endurance. So I wouldn't buy anything unless I had evidence that it was actually going to do anything. So, but I think it is an interesting. There's an interesting triangulation between performance, elite performance, the microbiome, and overall health. So I think that we will see more work coming on that.
Can you hear me? It was a real fascinating talk, Dr. Crine. You talked about how, when it comes to genetics, we can blame our parents and our grandparents for our genes. So similarly, what have you observed when it comes to microbiome? There's a lot of it also, very, very strongly correlated with what our parents give us. And just relating to that, so when, let's say, you introduce some microbiome to treat or to improve your wellness, does it have lasting impacts if inherently there's a different microbiome?
Great questions. So we get as I mentioned, we get our microbes from our moms. So that's the vertical transmission. It's not genetic. It's just a vertical passing over, and that there is some genetic. There's two elements of genetics that are really important. One is the genetic of the host. So genetics play a really important role in basically. I like to refer to it as where the microbes are housed. So basically, on making up the cellular context of the villi, that's all under genetic control. So indirectly, then that will affect the composition of the microbes, because you inherit the genes that are important for that. Same with the genes involved in immune senescence and various other aspects of immune function that will be very relevant in that. And on top of that, then we know certain genes, certain microbes themselves have kind of SNPs and polymorphisms that are actually passed on overall that might have some health benefit effects. Twin studies thought that the heritability was higher than it actually is turning out in much larger cohort studies. So I think it's an interplay and an important one, but probably not as strong as we originally thought. And environment. Your question about how long does it last? The environment is what's driving things back. And so if people change their microbes and their environment, then there's an opportunity then for it to persist. But if they stay in the same environment, then it reverts back.
The organisms that make up your microbiome are the majority or the most influential or whatever in terms of these brain acts. Are they prokaryotes or are they eukaryotes?
So the most studied are the bacteria. So in terms of how that's by far the most studied overall. There are single cellular eukaryotic organisms as well that will be playing a role, but they're not as well studied.
Back there.
Yeah. It's working, right? Yeah. So two short questions. One was that at some point you mentioned that just like so I'm not a biologist, so I might be saying something wrong. Just like the brain has sort of critical periods for development of various parts of it, and beyond that, it's hard to, if you lose something during those phases, then it's hard to get it back to that phase. And you said something like the microbiome also seems to have some critical phases. And if they do have critical phases, then an intervention like the one you mentioned, where you take something from the younger ones and give it to the older people, and that affects the microbiome. How does that effect quite work if there was a critical period beyond which it's hard to affect the microbiome?
Yeah. Great question. I could answer, we don't have all the data, but the key part here is that you perturb the microbiome in early life, and over time it will reverse. The environmental pressure will push it back to normal, but basically the horse is bolted. And so it's already caused changes to the brain overall. Now, the question about reversal, we know from other studies that perturbations to the brain, even though there's critical windows, but interventions later in life can reverse the effects of, say, early life stress or early life trauma overall. And in the same way, we think that even though, and in our study, interestingly, we didn't shift the microbiome completely back to where it was in young, we just changed it in a different way. So it could be a parallel pathway that could be applied to be able to reverse the effects. But it's a good question.
Thanks. Just one quick question, and I'll pass the mic. How optimistic or realistic is it to expect antidepressants based on these sort of bacteria?
Yeah, like we would hope that some of the interventions would already be like the studies are now emerging on diet. That the modified Mediterranean diet is having an adjunctive treatment in depression, so adding it on to antidepressant treatments. I'm not sure these will ever replace antidepressants or psychotherapy, but I think there'll be a really good adjunctive treatment. We need clinical trials, and they're expensive, and people don't want to do them. Because the cost isn't there, and we're dealing with a pharmaceutical model of how we develop medications for a food substance that just doesn't have the money behind it right now. So there's some regulatory and cultural aspects there that are really important to acknowledge overall. But I would be optimistic, but I'm that type of person.
Hi, can you hear me? Yeah.
Yeah.
One question on these, the new drugs on the block, the GLP-1 inhibitors, how they interact and what they do to the microbiome. And the second question is really, is there a shortcut to just getting the perfect microbiome with these probiotic tablets that we seem to get all the time?
So the first question, with GLP-1, there isn't any really good data yet that I've seen on let's say, Ozempic and the microbiome, but there's a feedback loop on metabolism. So like basically I think the microbiome is like they're like your internal Weight Watchers. So they're sensitive to any change in metabolic activity. We know the opposite happens, that the microbiome can cause GLP-1 to be released. So that has been studied. And I think that the feedback loop then, if you start to lose weight or whatever, then your microbiome will change and then try and recover on that. But we need more studies. The second question was, I didn't capture.
Magic bullet?
Magic bullet. There probably is a magic bullet for you, but it won't be the magic bullet for the person beside you. And that makes it difficult to be a magic bullet overall.
Clinical perspective, basically what we've noted is, you've been talking and I've been prescribing specific prebiotics, right? Or probiotics. And what I've realized is we've narrowed it down. I mean, we generally pills will be prescribed will be lactobacillus, will be bifid, bifidobacteria. Maybe now we're seeing signs of Saccharomyces boulardii coming in. But how comfortable are you with these specific organisms versus a relative diversity?
Yeah, it's a great question. I'm always a little bit cautious because unless there's data on that specific strain of bacteria, I think then we don't know really what we're doing overall. There's huge heterogeneity, and this is not what people really want to hear, but between one lactobacillus strain or one bifidobacteria strain, there's a huge difference. This was a learning curve for me some years ago, but as a pharmacologist, it makes sense. So a bifidobacteria longum is not just a bacteria longum. There's all various subspecies of that. And so unless we have data with human trials showing efficacy on whatever your readout you're looking at, I think it could be nothing more than a good placebo. And I would say maybe try diet if you can overall. But there are studies, there are some products where there is data, and then I would steer towards them. I don't know what's available in India, but there are in Europe.
Hello, sir. So really interesting talk.
And I was just wondering that, we've been talking about fecal transplant since a while now. And I'm really interested in the field of neurodevelopment disorders like ASD or ADHD. So how do you think the fecal transplant could help these candidates? Because currently, recently I read that UK noticed a shortage in medication. And I think that is a serious problem if we are talking on a larger scale with more people getting diagnosed now.
Sure. There are some studies, small open label studies with enriched consortia that are mimicking a fecal transplant and have shown success in ASD up to 4 years overall. It hasn't reached mainstream because of all of the hurdles with doing fecal transplants. They're widely done in gastroenterology, but outside of gastroenterology, it is quite hard overall. In relation to ADHD, there hasn't been much in that regard at all. I remain a little bit more optimistic that maybe through diet, and we're going to start a study now with our psychobiotic diet in adult ADHD. So just to see if it's going to have beneficial effects for attention and subdomains there. I think there might be other strategies to help some of the GI issues that are also common in ASD as well. But it is, it is a population that's looking for more solutions for sure.
I think as well with psychobiotics, as you mentioned, we could intervene with ADHD. I'm not talking about completely curing it, but probably managing the symptoms.
Yeah, exactly. And I think that's where that's where we go. And it's really important that people understand that that's not the goal at all. It's really about supporting optimal aspects of life.
Thank you so much.
We'll take the last question, and then there's one back there. I know there are so many questions, but perhaps we can catch John over tea. And I think as he heads down, maybe we can catch him for a few questions. But let's end this question now.
But you have to get your mom to ask a question.
Okay, but there's one more up there, and then I'll hand it over to my mom. Sorry. Yeah.
Like you said that the microbiome helps in the evolution of the organism. So have we ever in the research field tried to compare the microbiota in the astronauts or the people living in the say ISS and to those of the Earth? And are there any type of significant changes occurring in their microbiome?
Yeah, people have done this, and they've also done it in the twins that went up to space and looked at their microbiome and looked at it also in relation to mental health. There was one study out of China some years ago, which looked at 4 people training to go up to space who were in complete isolation, and there they correlated the mental health with their microbiome across the time window as well. So this is right, but it's small numbers. So it is small numbers, but it is relevant.
Last 2 questions, and then really genuinely last 2, one here and one there.
Wonderful talk. I enjoyed it. You did mention about sleep and microbiota. What happens to the social jet lags, professional jet lags, and those who are really compulsory, like flyers, pilots and things like that?
Yeah, so people are beginning to study that now really, really well. There was one study out of Israel where they looked at people as they crossed the Atlantic and collected. They also looked at people collected globally. They poop from a flight before and after, and were able to do all these types of studies. So it looks like that there definitely is changes in the microbiome. And I didn't have time to go into it there, but our data is now supporting that it's not only what we feed our microbiome, but when we feed it. So, that's really important. And that may also have implications, not just for social jet lag, but also for things like fasting and other aspects of life that are really important there as well. But it's a really great question, really, really great question.
Hello. So you mentioned that the brain gut axis, it regulates craving in young binge drinkers. So I've heard of a disease named pica in which individual craves non-edible substances. They have a very constant craving of non-edible substances, and I've heard that doctors have not really found the real reason for it. So would it be related to that, the cravings?
I didn't understand. So in the binge drinking?
Like.
Sorry?
non-edible substance injection and whether that is linked in any way.
Oh, okay. So kind of an obsessive, yeah. So we have no data, like as you would expect, no data yet. But from a brain perspective, and from a circuit perspective, and from an understanding about repetitive and obsessive behaviors, and the microbiome is involved in everything because the microbes were there first.
Okay, so let's thank John for a fantastic talk.
And there's tea outside, so please help yourself.
Thank you. I'm okay. I'm okay. I'm okay.